Perfect. So then, my name is Johan Bengtsson-Palme. I am an assistant professor at University of Gothenburg, but I have actually spent most of my time at Chalmers University of Technology, which is the technical university in Gothenburg since 2022. So just to shout out to Chalmers there as well, so we don't forget about them. I will actually start in this end so that they are not forgotten. This is the fantastic people who have been performing the research I will be talking about today. This is my research group in sometime during the winter. My research group is working with a little bit, maybe 2/3 are working on antibiotic resistance, primarily on antibiotic resistance in the environment, which I will talk about today, but we also do quite a bit of work on microbial interactions and how microbes talk to each other and competitive interactions, collaborative interactions in microbial communities. But that's something I'm not going to mention much today at all. So this is a slide that I think everyone in this room has probably seen a number of times. It's also fairly old. It basically shows the number of deaths of different causes and try to put antibiotic resistance in context. And then here it says that in 2016, 700,000 people would die every year from antibiotic resistance. And unfortunately, the projection for 2050 is not looking much better today. We're still looking at probably 10 million people per year dying from resistance-associated infections. And just to put that a little bit in perspective, 10 million people dying annually from antibiotic resistance would essentially mean that everyone in this room would know someone who dies from resistance every year or every second year, something in that range. So it would be one of those really common things that we hear about almost daily, because someone will know someone who dies from a resistant bacterium. So that's the magnitude if we can't control the problem. So what my research group is trying to figure out is, can we prevent this somehow? Is there something we can do to help prevent this resistance catastrophe that we have? Everyone would have to know someone who dies from resistant bacteria every year. And of course, we're not going to solve the problem ourselves, but maybe we can contribute a little bit to that solution. And where we specifically try to look is in the environment, and not really because I think the environment is necessarily the most important place to look for antibiotic resistance, although I will come to why this could be important. But largely because there's already a lot of effort being spent in, for example, resistance in clinical settings, in agricultural settings, and the environment has at least traditionally been somewhat overlooked. And I still think that this could be potentially very important. And I will get back to exactly what the reason why I think that is important. Well, we'll start in this end. This is from a paper we published about 5 years ago, where we tried to model, if we think about the resistance genes that have emerged in pathogens in the past 75 years, where is it likely that these come from? Based on the data we have on how resistance genes move, on the selective coefficients of resistance genes, on how many resistance genes we do find in current pathogens and so on. If we try to model the process of resistance genes emerging and ending up in pathogens, where is it likely that they would come from? And we looked at a few different origins. We looked at if they could come from, do you see my cursor if I do this, by the way? Yes, good, then I can point with that. So we looked at, for example, is it likely that the emerging pathogens that are already in humans or in non-pathogenic human-associated bacteria would have been on mobile genetic elements like plasmids? Could it be like they are on opportunistic pathogens in the environment? Could it be that they are on the chromosomes of environmental non-pathogenic bacteria, or could it be on mobile genetic elements in environmental bacteria? And we tried to look at how likely is it that the average resistance gene that we find in a pathogen today would have had any of these origins. And this is a summary graph, and I'm trying to break it down for you, break down the results of the entire study for you here. So what we basically come down to is that it depends a lot on what assumption one makes on what has happened in the past 75 years. How much do we think that it costs for a bacterium in terms of fitness cost to carry a resistance gene? How easy do we think it is for the resistance gene to move from the chromosome onto a mobile genetic element? How often do we think that mobile genetic elements spread between bacteria and so on? So depending on what assumptions we put in, we get very different results, and that's reflected in these bars that are extremely wide. This is basically a likelihood that we're looking at here. So to make this story short, the current resistance genes that we do find in pathogens seem slightly more, according to this model, likely to originate from human-associated bacteria than environmental bacteria, but only slightly. It also seems really important to be close to humans, like having a short dispersal route from environmental habitats to humans. So even if these resistance genes have emerged in the environment, it has to be in environments that have close connections to humans. It also seems to be crucial to get onto a mobile genetic element early in this process. Otherwise, the resistance genes tend to not be widespread in pathogens. But the main point that we make in this paper is that it's all very uncertain. We have way too little data to be able to tell with any kind of certainty where antibiotic resistance genes have actually originated. It is actually possible within the model parameters that every resistance gene was in every pathogen 75 years ago, and then we have just had counter selection against them. But it's also possible that they have only been in basically a single environmental bacterium somewhere. And that means that since both of those scenarios would be possible under the model conditions, it basically means that the uncertainty is huge. And the short story is we don't know where resistance gene come from. We still don't know. There are some indications on that the environment is important, though. So this is not our study. This is a really nice paper published in Nature from 2011, where they looked at these 30,000-year-old permafrost samples that have been untouched by human hands. And they sequenced these permafrost samples, and they find the same, they find DNA from mammoths, they find DNA from bisons, but along this DNA, they also find the same resistance genes that we have problems with in the clinics today. So these resistance genes have apparently been there 30,000 years ago. And of course, it's very much possible that the mammoths had a very advanced healthcare system with a lot of antibiotics being used, and that's why we find them. But the more probable reason is that these antibiotic resistance genes have been there for thousands, probably millions of years, and have either functions in protecting bacteria from other microbes or completely different function, and they turn out to be resistance genes when overexpressed, for example. Similarly, there are a study by Forsberg et al., which looks at resistance genes in pathogens and soil bacteria, and they find these 100% identical resistance genes in pathogens and in soil bacteria, despite that the rest of the genome outside of the resistance gene is very different. But still, the resistance genes themselves are identical, which basically is a strong indicator of a recent transfer of DNA between pathogens and soil bacteria. This is a study from 2018 where I've been involved, where we looked at soil samples from around the world, dominantly from Estonia and northern Sweden, though. And we basically look at a lot of different things, but the thing that is important for this presentation is that we look at these antibiotic-producing fungi, and we compare that to the total antibiotic resistance genes in the same sample, and we find this positive correlation, pretty strong correlation, which is again an indication of that if you have more of these antibiotic-producing fungi, you have more resistance genes, and potentially that could also be then the reason that you have more resistance genes, that you have antibiotic-producing fungi that create a selection pressure for antibiotic resistance genes. So all of this points to that the environment can have an important role in the selection of antibiotic resistance. And we wanted to just look at this a little bit more directly. Can we look at exactly just selection for resistance and not introduce a lot of other parameters? So we have done a study where we have been looking at adding ciprofloxacin at low concentrations and environmentally relevant or above environmentally relevant concentrations, and tetracycline at environmentally relevant concentrations and way above the relevant concentrations. And we've just been exposing soil samples for 1 day or 8 days, done shotgun sequencing and then metagenomic analysis to see if we can detect an enrichment in a resistance gene in a relatively short-term experiment. And what's interesting in this study is that we actually don't see that much happening for the relevant antibiotics. So I've just pulled out the resistance genes here that are actually resistance genes against ciprofloxacin or tetracycline. And as you can see, there is not a lot happening in these samples during or after the exposure. We do see some enrichment of specific taxa, for example. We do see some enrichments in non-associated antibiotic resistance genes. We do see some movement in mobile genetic elements, but there doesn't seem to be any specific selection for the resistance genes against fluoroquinolones and tetracyclines, which are the antibiotics we expose these soil samples to. So our conclusion here is that it's a little bit complicated to create these kind of environments where you can select for antibiotic resistance, where you can use antibiotic selection only and like break down this is the antibiotic selection component. Because what usually happens is that you add antibiotics together with, for example, manure on soil, and then you also add a lot of nutrients. You also add a lot of other bacteria derived from feces. So you're basically adding a mix of things. But when you try to just look at the selective component itself on soil bacteria, we don't see much happening, at least not in the short term. And this also, agrees pretty well with a much longer term, years-long experiment in southern Sweden, looking at adding antibiotic-containing manure for tens of years, and also seeing very, very minute or even no effects at all in the long term, even though we can detect these short spikes just after adding the manure. So still, we do find quite a lot of resistance genes in the environment. So that means that we would like to understand what is the normal type of resistance we actually encounter. So in a study by my postdoc, Anna Abramova, we looked at in different studies, qPCR studies, and what they detect in terms of resistance gene abundances in different environments. The goal here was basically to try to identify, first of all, what is the normal background levels we find of resistance in different environments, and second of all, are there specific targets that would be particularly useful for indicating, for example, human anthropogenic impact or selection with antibiotics. So the bottom or the top line results of this is that the average resistance gene in the average environment is present in 1 out of 1,000 bacteria, approximately. So that's the thick black line you see in this figure here, which a peak around here at 10 to the power minus 3. So that's the average resistance gene in the average environment having a typical abundance of 1 or existing in 1 out of 1,000 bacteria. But what is interesting is that, first of all, you can see that there's a lot of different distributions for all the different genes. So different genes have very different typical mean values in different environments. And what's also interesting is that we can, at least for the more common resistance genes, break these down into likely unimpacted, likely impacted, for sure impacted, and feces environments. So TET-A here, for example, you can see a clear difference between the human impacted environments here, which have higher abundances, and the non-impacted environments down here, which have substantially lower abundance of the TET-A resistance gene. So this is a good example of something that could serve as a marker of human pollution. Similarly, the TEM beta-lactamase also has a clear separation of these distributions, and so does the INT-I integrase, which has been often proposed as a marker of anthropogenic pollution, although the INT-I integrase is actually a worse marker than, for example, the TET-A resistance gene would be. So this is something interesting that came out of this, and also we could start looking at how this was actually distributed within different environments and say something about what would be normal in a specific environment for this resistance gene. So for example, you can see that the sediment distribution of TET-A here is in this range, and then we have these two clear outliers. So this would probably be an indication of a sample that you would like to investigate further, while everything that ends up in this range here would be more normal in terms of background levels of antibiotic resistance. So we think this could have pretty good uses in environmental monitoring of antibiotic resistance. It also allows us to build these kind of global antibiotic resistance maps where we can say something about how common are certain resistance genes in certain parts of the world. But one thing that really came out of this study is that our current picture of this is extremely biased. So we have a lot of data from Europe, we have quite a lot of data from North America and Southeast Asia, but there's almost nothing from Africa, and there's no qPCR studies at all that we could use the data from in Latin America. So the picture we have of antibiotic resistance globally is very biased, and it's very biased to countries that have generally better sanitation standards, for example. So we know very, very little about what the environmental antibiotic resistance situation is in South America and in Africa, for example. That was also something that came out, maybe not as a surprise, but the magnitude of that difference was a surprise to me. So I talked a lot about antibiotic resistance genes in the environment and how they might be moving around and being selected for. But one of the more ignored aspects of the environment is that, as opposed to, for example, the human microbiome, where it's likely that most of the resistance genes that can be transferred to pathogens have already been transferred to pathogens because the distance between human-associated bacteria and human pathogens is really short. I mean, it's actually pretty likely that a lot of the resistance genes that have not already been mobilized into pathogens are not compatible with pathogens when it comes to human microbiome. But the difference with the environmental microbiome or environmental microbiomes, because they are very diverse, is that it's huge. And there's a lot of bacteria there that have probably never really had the chance to transfer their genetic material to human pathogens. So we still think that there is a huge reservoir of potential novel resistance genes that could function well in human pathogens, even though they haven't gotten there yet. And in many cases, those resistance genes might actually be more concerning because the ones that already circulate among human pathogens, they will keep circulating among human pathogens. If you have a transfer event from one pathogen to another, it has some degree of impact on the treatability of the infection. But if you have a new resistance gene coming in from an environmental bacteria that haven't existed in pathogens before, and that can start spreading, it has the potential to have very small impacts because the resistance gene is not very efficient. But if it's an efficient one, it could really turn the entire treatment possibility situation upside down. So what we're thinking here is that it would be really important to understand these resistance genes that have not yet come from environmental bacteria into human bacteria to better anticipate which of those should we potentially worry about. So we're working on a few different ways to try to discover these new forms of resistance genes. One of them is something called transposon mutagenesis, where we basically can use a transposon that randomly inserts itself into different genes of a bacterial genome, and then we can use a clever PCR and sequencing protocol to quantify the impact of this disruption that the transposon does on a large scale. I'm not going to go through the details here, but what this allows us to do is to, for example, expose a bacterium like Pseudomonas aeruginosa in this case to an antibiotic and see which of these transposon-generated mutants have worse fitness in the face of an antibiotic. So which ones would be necessary to be able to survive the antibiotic. And when we do this in Pseudomonas aeruginosa, we expose to tetracycline and ciprofloxacin at 2 different levels, we pick up a number of genes that are important for the tolerance to these 2 antibiotics. So all of this is at sublethal levels. But what is interesting is this overlap here, for example. We have a bunch of genes that are important for tolerance to both tetracycline and ciprofloxacin. And we thought those were particularly interesting because they might have some kind of multi-resistance function. So what Emil Burman, my previous PhD student who performed this, did was that he took some of these genes, which we have indicated in the red boxes here, and did follow-up experiments after this high throughput experiment to try to determine if they actually became more susceptible to the antibiotic when we deleted these genes. And sure enough, for every single gene we picked up in this study, we could detect some degree of increased susceptibility to the antibiotic when we did follow-up experiments. And if we look at this non-related gene, SIL-L, which had no indication of this, we just picked it up because it was important to have something to compare to. We wanted to see that it's not just that we're deleting a gene. We see that pretty much all of the genes that we picked up in this screen have some kind of impact on fitness when you remove them. So what we would like to see, of course, now is that if you overexpress these genes, they would give more resistance. Unfortunately, that's not really the case for any of these. This one over here has a little tendency in that direction, but it's not a particularly strong effect. So essentially, it looks like what we're looking at here is genes that are important to tolerate antibiotics, but if you overexpress them, they don't really gain any additional resistance. So this might not have been that great of a method to discover new antibiotic resistance genes, even though it was nice to see which genes infer antibiotic tolerance in Pseudomonas aeruginosa. So we tried something else, and it turns out we don't have to go that far out in the environment to discover interesting antibiotic resistance genes. So this is actually me sampling sediments from one of the local parks in Gothenburg. And what we did here is that we took these samples, we brought them into the lab, we exposed them to the beta-lactam ertapenem for 7 days, and then we looked at phenotypically if we have selected for more clinically resistant ertapenem, or for more bacteria that carry clinically relevant ertapenem resistance, and we also did nanopore sequencing to see if we could discover new gene variants connected to this. And we did nanopore sequencing of both the sediments and also the plates where these resistant bacteria grew. And through this approach, we could identify a bunch of novel and potentially novel beta-lactamases belonging to the class A, class C, and class D. So specifically, I think that these are interesting. This one here is a class A beta-lactamase that we followed up, and I will come to that. But the ones that I marked here have been selected for follow-up studies to verify that they are actually conferring ertapenem resistance. So this one here, we also have a few over here, and I don't think there is anyone in class D, no. Okay, so those we've selected, and you can see here that these are also related to known beta-lactamases in some cases, and in some cases they are quite distant from other ones. So if we look at how similar these are to known beta-lactamases, it turns out that all of them are in the 50 to 65% identity range to known beta-lactamases in ResFinder. And when we did verification studies of these, we could see that pretty much all of them, aside from perhaps number 14, give clinical levels of resistance. So the clinical level of resistance is here. This is NDM1, which we included as a positive control. We also have this one, which only gave clinically relevant levels of resistance in one of our tested constructs. So basically what we've been doing here is that we've been cloning these new resistance genes into a plasmid where we can overexpress the beta-lactamase and see if it's actually functional. What's interesting is that we can use these exposure experiments also on stored samples. So I'm not going to dwell on this because I see that I'm running out of time, but we can actually store samples in the freezer for 6 months and still get relevant results out of this. And this opens up a lot of interesting things in terms of bringing samples in from interesting environments in other countries and being able to expose them also after being frozen. It also opened up some opportunities to go back and do selection screens on historical samples, which I think is interesting. Through this method, I mean, here we've only looked, what I've showed here was only beta-lactamases, which is, it's likely that we will find beta-lactamases when we expose to meropenem. But it's actually, this type of experiments also has the potential to find completely new antibiotic resistance gene classes that are not structurally related to something we already know. So this example here is a tetracycline exposure experiment on biofilms, where we have blindly quantified genes that are enriched from metagenomes exposed to increasing concentrations of tetracycline. And when we have picked out these enriched genes and annotated them, we find a bunch of known tetracycline resistance genes, primarily TET-G and TET-C. But what is really interesting is that we can also pick up other genes that have the same pattern. And those genes are not annotated as tetracycline resistance genes. So we can see that we have this TON-B dependent resistance. We have two different transporters. We have a gene called TOL-B, and we also have this completely hypothetical protein that doesn't have any annotated function in the databases. We don't know for sure if these are tetracycline resistance genes or if they're somehow managed to just hitchhike along in these experiments. But we will try to express these as well in our overexpression system and see if we can also see a resistance phenotype from any of these genes, which then would allow us to actually go outside of this homology related prediction of resistance and actually make it possible to pick up resistance genes that are completely non-homologous to known resistance genes. So one question we have also been asking ourselves is like, all of these resistant genes that have not yet been established in pathogens that we call latent resistance genes that are waiting to potentially be transferred to pathogens, where do they actually come from? Where can we find them? And this is a study I did quite a few years ago, comparing these resistance genes from pathogens to resistance genes that have been identified in other bacteria using functional screens, for example. Built two different databases of that and compared the results on metagenomes. And what we find is that if you look at abundance alone, most known resistance genes from pathogens we do encounter in pharmaceutical polluted environments where they have been exposed to antibiotic, to waste water from antibiotic production. That's also where we find most of these latent resistance genes in terms of abundance. What is interesting, though, is that if you look at diversity instead, it turns out that the biggest diversity of these novel resistance genes we actually find in soil and water environments. So this means that there's a big diversity of genes that could potentially be selected for under the right conditions. We have seen that also in another study where Johan Bengtsson-Palme looked at where we can find predicted resistance genes. So this is computational predictions of antibiotic resistance genes based on existing gene classes. And we find that we have a lot higher diversity in basically every environment of these predicted resistance genes than of the established ones that are already in pathogens, although the abundance is not necessarily higher. And if we look at the abundance of these genes, we do see that the environment where you find the most of all of these gene classes is wastewaters and birds. So I guess if you don't take anything else from this presentation, don't eat bird poop could be something you can take home. Tess, I guess I should wrap up, right?
Yes, that would be great if you can come to your conclusions.
Yes. Then I will very quickly just take one more thing about our resistance genes that we found in Slottsskogen in the park, and then why that is an important point. So if we go back to those resistance genes, I said that these were about 50 to 65% similar to known resistance genes. But how can we know which ones of those are truly new, which have never been looked at before? I mean, I just talked about studies where you look at functional predicted resistance genes, functionally verified resistance genes that are not in the clinical gene databases. So how do we know that these are not in another database somewhere or have been studied in another data set? Well, basically, I'll skip this one and go straight to this. So when we start comparing to other studies, we realize that this one, even though it's only 50% similar to clinically detected resistance genes, is 94% similar to a gene found in this Indadias study I just talked about. So we can see that this gene has been studied before in another study, but since it doesn't have a common gene name, there's no connection. We can't say that this gene has been picked up before. And this highlights that there's a need for some kind of centralized repository for resistance genes, where these genes can be marked as either being established in pathogens, latent in environmental bacteria, and whether they are mobile or if they have been experimentally verified. And by cross-checking all potential novel resistance genes against such a resource, it would be easy to identify if a gene has already been assigned to resistance function before. And we can then know when we truly have done a new novel gene discovery instead of just rediscovering the same novel resistance genes over and over again. And in an effort to try to build this database, I have started something called Clever, which is a classification scheme for latent and established variants of extant antibiotic resistance genes. This is still in development, but there is an early version available at microbiology.sc/clever for people who want to check this out. I am not going to go through all the details, but for example, if you look at these two examples, here is an aminoglycoside resistance gene class. You can see that we have looked, these variants are the variants that already exist in ResFinder. These ones does not exist in ResFinder, but they have been picked up in ResFinder FG, so for functional metagenomics. And these three here at the bottom were detected in the DIA study, but has not been included in any of these databases. And we can also get indications on whether these are mobile or chromosomal, and whether they have been have a functional verification or are only computational predictions. This is the corresponding list of tetracycline enzymatic resistance genes, where most of them have already been found in ResFinder. Some of them are only in the CAR database, and then there's one additional one that was added only from the INDA-DIAS data. So this is something that I think will be hopefully very useful once it's finished from a development point of view. And we are already using this internally in my group. So with that, I'd like to thank everyone who's been involved, who has contributed, and who has been working with me in the past few years, along with all of our funders. And I'm sorry for dragging out a few extra minutes. Thank you.